Mrs.K.Vijayalakshmi, Assistant Professor, Department of Genetic Engineering, Bharat Institute of Higher Education and Research, Chennai 73, is a handle to the course name Cytogenetics, course code U18Pcge603. Topic is genetic inheritance. First of all, see about the prokaryotic cells. There is no membrane-bound organelles present there. Simple genome organization. Genes usually do not have introns. Between 500 and 4,000 genes. The case of Mycoplasma genitalium has 477 genes. Great degree of diversity. Units. There's a structure of prokaryotic cells having plasma membrane, inner side having DNA, and outermost layer is cell wall having flagellum for motility, and the ribosomes are present in the cytosol having 1 micron in size. Then DNA of prokaryotic cells. In prokaryotes such as bacteria, the bacterial chromosome consists of a circular double-stranded DNA molecule. More than one copy of the bacterial chromosome may exist in a bacterial cells. Since prokaryotes do not have a nuclear membrane, each bacterial chromosome is packed in the region of the cell called the nuclei. The DNA of a prokaryotic cell, such as E. coli, is packed near the center of the cell in the nuclei, which is not segregated by membrane from the rest of the interior of the cells. The prokaryotic DNA must be tightly packed so that it can fit into the nuclei. DNA packaging is achieved through coiling, compacting, and supercoiling. So the diagram represents A. The circular chromosomal DNA molecule can be compacted through the B represent the formation of looped structure. C. The looped DNA can be further compacted by DNA supercoiling. So note the colored ball represent protein involved in supercoiling. The most prokaryotes are haploid, meaning they only have one set of chromosome and therefore carry only one copy of each gene. Their genomes carry very little non-essential DNA. So the majority of prokaryotic genomes are composed of regions that contain either genes or regulatory genes. Regulatory sequences are sections of a DNA sequences that determine when certain genes and associated cell functions are activated. So this is a partial map of E. coli genome. So there are actually thousands of genes in the genome, very few of which are non-essential. As a prokaryotic DNA having origin of replication and terminus of replication. So in E. coli K, so 12, having 4,639, 221 nucleotide pairs. Next, eukaryotic cells. So our organelles are membrane bound, complex genome organization, large genome size between 6,000 and 30,000 genes. So genes have introns where the genetic diversity takes place. A large amount of regulatory DNA that control gene expression, a high degree of genetic redundancy, unicellular or multicellular. So this is a general eukaryotic cells having plasma membrane, intermediate filaments, Golgi apparatus, ribosome in cytosol, perioxysome, actin filaments, then microtubules, centrosome with a pair of centrosols, then chromatin DNA, nucleopare, nuclear envelope, then extracellular matrix, then vesicles, lysosome, mitochondria, endoplasmic reticulum, nucleus, nucleolus, and plasma membrane. The general structure of eukaryotic cells. So then say about the DNA organization in eukaryotic cells. The histones, a family of protein that associated with DNA. Nucleosome, the condensed structure formed when double-stranded DNA wraps around an octamer of histone proteins. Note that organization as a distinct chromosome is shown in D, capital D, occurs during cell division. So otherwise, the DNA is less condensed and is called chromatin. Then chromosomes in eukaryotes that are found in the nucleus, condensed and visible during cell division. So at the beginning of mitosis, they can be seen to consist of two threads, nothing but sister chromatids, joined by a centromere. The sister chromatids are identical copies. So during mitosis, the sister chromatids separate and are placed into two nuclei. This is the chromosome structure. There's the structure of chromosome having if you see the one chromosome having two identical chromatids. One is an exact copy of the other and each contain one DNA molecules having P and Q arm. P arm is a short arm structure. Q is a long arm structure in center for centromere that consistent point of the chromosome. Then within the chromosome, the DNA molecule presents a long string-like DNA molecule formed into a compact structure by a protein called histones. You see about the numbers of chromosomes that are constant for each cell in the body except sex cells which only have half sets constant together through the life of an individual. You don't lose or gain chromosome constant for all members of a species. There's a chromosome present in mouse and in mice. Then chromosomal numbers seen in the humans are 46, chimpanzee 48, house mouse 40, mice 20. This is a human chromosome. So how to identify the chromosome means by their size, their shape, nothing but the position of the centromere, then the banding pattern produced by specific strain, nothing but genes are strain. So the chromosomes are analyzed by the organizing them into a karyotype. Thus, development and chromosome. So, differences in chromosomes are associated with differences we can grow. The karyotypes of males and females are not the same. Female have two large X chromosome. Male have a large X and small Y chromosome. The X and Y chromosomes are called sex chromosome. The sex chromosomes are placed at the end of the karyotype. So the unusual growth can be associated with chromosome abnormalities. Example, people who develop Down syndromes have trisomy 21. This is a chromosomal abnormality seen in Down syndromes having trisomy 21 and in Edwards syndrome trisomy 18. Next, see about the sex chromosome. The sex of many animals is determined by genes, but on chromosomes called sex chromosome. The other chromosomes are called autosomes. One sex is a gamete, and the other sex is a heterogamete. So you see difference between the chromosome in eukaryote and prokaryote. In prokaryote, single chromosome plus plasmid is present. In eukaryote, many chromosomes are present. And the chromosome in prokaryote are circular. In eukaryote, linear chromosome. In prokaryote, the chromosomes are made only of DNA. In eukaryote, made of chromatin, a nucleoprotein, nothing but the DNA coiled around histone proteins. In prokaryote, the chromosomes are found in cytoplasm. In eukaryote, found in a nucleus. And prokaryote, the copies its chromosome and it divides immediately afterwards. In eukaryote, copies chromosome, then the cell grows, then goes through the mitosis to organize chromosome into equal chromosome. Next, we see about the genetic inheritance. So humans have 23 pairs of chromosome. Each pair of chromosomes are what we call homologous, meaning that they contain the same genes. So therefore, everyone has two copies of every single human gene. Fail-safe mechanism in case one gene in a cell fails, there is another to carry on. So 23 of these pairs are called autosomes. One pair are called the sex chromosome and determine the sex of the individual. In male having XY chromosome, female XX chromosome. So humans have 23 pairs of chromosome. 22 of these pairs are called autosome. One pair are called the sex chromosome and determine the sex of the individual. So in human having this XX chromosome, if the one X chromosome having a mutation mean that will not cause the disease, but the two X chromosome undergo mutation mean that lead to the disease. But in the case of men having only one X chromosome, if it muted mean that lead to produce a disease. So having this female karyotype 46 XX chromosome, then the male karyotype 46 XY chromosome. Then the gamete formation and function. So what is gamete? Is a gamete is a haploid sex cells which has to fuse with another sex cells of the opposite sex in order to survive and pass on its gene to form a new individual. Then what is fertilization is the fusion of two haploid cells to form a single diploid cell called the zygote. Then allele, so an allele is a particular form of a gene can be represented as a letter, for example, capital S or small s, capital H or small h. Alleles are the different forms of the same gene. Example, capital S is the dominant form of the gene, whereas S is the recessive form of a gene. The locus, the plural loci of a gene is the position in is occupied on a chromosome. And genotype of an organism refers to its genetic makeup. Example, capital S, small s. The phenotype of an organism refers to the physical appearance or characteristics of that organism. Example, capital S, small s can be responsible for a physical appearance or characteristics. The genotype and the environmental conditions together have an effect on the phenotype. First, you have the allele and loci before meiosis. You take the male heterozygous for brown eye and female heterozygous for brown eye. So after meiosis, that shows the mental first law of segregation. Then alleles and loci after meiosis, it produces a sperm and eggs. In the case of sperm, half sperms are capital B and half are small b. In the female heterozygous brown eye, the eggs having half eggs are capital B and half eggs are small b. So allele and loci after the fertilization. For example, male heterozygous for brown eye and female heterozygous brown eye. After fertilization, you can get this phenotype of F1 generation is blue eyes. So genetic inheritance, alleles of the eye genes. B is responsible allele for brown eye, so dominant to the small b allele. Small b allele for blue eye, recessive to the capital B allele. So for possible genotype is, for example, homozygous genotype means that capital B lead to corresponding phenotypes as a brown eyes. If the genotype is heterozygous, for example, capital B small b lead to corresponding phenotype as brown eyes. If the genotype is homozygous, nothing but small b lead to corresponding phenotype is blue eyes. So genetic is a study of inheritance to the first filial generation, nothing but F1 generation of a single unlinked triad in a cross involving homozygous parent, heterozygous parent, then sex determination. First, you take the homozygous parent, nothing but homozygous brown eye, capital B, homozygous blue eye, small b. The gamete formation is capital B, small b. The F1 progeny genotype is capital B, small b, capital B, small b, capital B, small b, capital B, small b. So the F1 progeny phenotypes, all offsprings have brown eyes. If you take the heterozygous parent, heterozygous brown eye, capital B, small b, heterozygous female, capital B, small b. So F1 progeny genotype is capital B, capital B, capital B, small b, capital B, small b, small b, small b. So the F1 progeny phenotype is 3, ratio is 3 is to 1, 3 brown eye and 1 blue eye. To take the heterozygous and homozygous param, heterozygous brown eye, capital B small b, and homozygous blue eye small b. The F1 progeny genotype is capital B small b, capital B small b, small b, small b, small b, small b. So the F1 progeny phenotype, the ratio is 1:1, that is one brown eye and one blue eye. So the genetic inheritors of eye color, brown and blue eyes, if you put it in a Punnett square, you got like this. 3:1 ratio. So next you see about the sex determination. So why in humans the father determine the sex of a child means the female are having the XY chromosome, then female male we are having XY chromosome, female XX chromosome. So after meiosis, the possible male gamete is X and Y, the possible female gamete is X and X. So after fertilization, either you got this XX chromosome that represent the female, or XY chromosome that represent female, male. So next we see about the incomplete dominance. So incomplete dominance is where two homologous alleles are equally expressed and neither allele is dominant over or recessive to the other. So the heterozygous genotype produces a phenotype intermediate between those produced by the two homozygous genotype. So an example is flower color in Snapdragon. Red flower crossed with white flower produces pink flower offsprings. So you see this incomplete dominance. The parental phenotype is red flower and white flower. Parental genotype, in the case of red flower, capital R, white flower, small r. So gamete genotype is either capital R, or small r. So possible fertilization, the gamete genotype is capital R, small r. So that represent the phenotype pink, and capital R, small r, that represent pink color flower. So Gregor Mendel. So Mendel studied the inheritance of 7 characteristics of pea plant. First one is sheet shape, second seed color, third ripe pod shape, unripe pod color, flower position, flower color, height. So these 7 characteristics were chosen because each has only two clearly contrasting qualities. First one is sheet shape, round or smooth versus wrinkled, seed color, yellow versus green. Then third one, ripe pod shape, inflated or versus constricted. Fourth one, unripe pod color, green versus yellow. Flower position, axial versus terminal. Flower color, purple versus white. Then height of the stem, tall versus dwarf. So Mendel used pea plants because they have several advantages over other plants. So they have a short life cycle, relatively easy to cultivate, could be grown in large numbers, are capable of self-pollination and fertilization. So Mendel used pea plants because they have several advantages over other plants. They have a short life cycle, relatively easy to cultivate, could be grown in large numbers, are capable of self-pollination and fertilization. So Mendel developed separate population of pea plant, each a pure breed, nothing but homozygous for a particular quality. Example, for height, Mendel developed pure breed homozygous tall pea plant and pure breed homozygous dwarf pea plant. This took a long time to achieve as Mendel had checked that the pure breed tall plant always produce 100% tall offspring, and ditto for dwarf pea plant. So Mendel kept strict record of his results. Mendel converted the results of his many crosses into a simple ratio that gave him an insight into mechanism of inheritance and lead to his two famous law of genetics. So Gregor Mendel's first cross, Mendel carried out two consecutive crosses. The phenotype is tall and dwarf. Genotype for 12 capital T, dwarf small t, the F1 generation having this capital T, small t, that cross with capital T, small t, they undergo self-fertilization. The F2 generation having three tall plants, capital T, small t, then one dwarf plant having this genotype, small t. So Mendel's first law of genetics, law of segregation. So he repeated this cross for the other six characteristics of a pea plant and consequently came up with the same ratio 3 is to 1. So the Mendel worked backwards and came up with the law of segregation. So what is law of segregation is each cell contains two factors for each triad. These factors separate during the formation of gametes so that each gamete contains only one factor from each pair of factors. So at fertilization, the new organism will have two factors for each triad, one from each parent. Let's see about the dihybrid crosses. Having worked out the mechanism governing the inheritance of one characteristic, Mendel then proceeded to study the simultaneous inheritance of two different characteristics. Example, height and seed shape. So again, Mendel began his dihybrid cross with the pure breed of the characteristics he wanted to study. So Mendel knew from his monohybrid crosses that tall and round seed are dominant, and so dwarf and wrinkled seed are recessive. So the phenotype is tall, round. The genotype is capital T, small r, dwarf, wrinkled, is a small r. So gamete is capital T, capital R, small t, small r. So first generation is tall, round. The genotype is capital T, capital R, small r. So Mendel understood the result of the F1 generation from a cross of parents homozygous dominant and homozygous recessive because the only offspring that could be produced from his cross was heterozygous individual because each individual could only produce one type of gamete due to the fact that they were homozygous for the two traits studied in this cross. The hybrid cross between the parent plant heterozygous for both traits was a problem. How are the gametes made from the genotype? Nothing but capital T, capital R, small r. So mental solution to the problem of gamete formation involving more than one characteristic is Mendel's second law. Nothing but the law of independent assortment. So law of independent assortment that discusses members of one pair of factors separate independently of members of another pair of factors at gamete formation. So next see about the independent assortment. The parent is tall, round. The crossed with tall, round. The gametes are capital T, capital R, capital T, small t, capital R, small t, small r. We have got this out of 16, you got 9 tall, round. Out of 16, 3 are tall, wrinkled, 3 are dwarf, round, and 1 is dwarf, wrinkled. So note that the tall, wrinkled, dwarf, round, and dwarf wrinkled progeny are called recombinant because they differ to the parental genotype as well as the phenotype. We cross this tall, round, and dwarf wrinkle, got the gametes capital T small r, capital T capital R, capital T small t capital R, small t small r. So out of four, one is tall round, one is tall wrinkled, one is dwarf round, one is dwarf wrinkled. So note that the tall wrinkled and dwarf round progeny are called recombinants because they differ from the parental genotype and phenotype. Next is see about the non-linked versus linked. The genes governing the traits studied by Mendel were found to be non-linked, meaning that each trait studied was on a separate chromosome to another trait. So note that non-linked genes are on different chromosomes and so will undergo independent assortment and therefore are true to Mendel's second law. So linked alleles or linkages, the linked alleles are those genes found on the same chromosome. So example of linked genes, in the case of mice, capital C, nothing but the colored seed is dominant over the small c, nothing but colorless seed, and capital S, the full seed is dominant over the shrunken seed, small s. So firstly, a heterozygous colored full seed, capital C small s, capital S small s mice plant is crossed with a homozygous recessive colorless shrunken seed, small c, small s mice plant. Secondly, two heterozygous colored full seed, capital C, capital S small s mice plant are crossed. So note that the genes for colored seed and full seed are linked tightly. So linked genes are the genes that are present on the same chromosome. Note that genes are said to be tightly linked if they are close together on the same chromosome. Tightly linked genes tend not to follow the mental second law of independent assortment. This is this non-linked genotype, capital R, capital T, small t. The linked genotype, you see the picture, capital R, capital T, small t. So parent phenotypes is a colored full crossed with colorless shrunken. The parent genotype, capital C, capital S, small s, crossed with small c, small s. So after meiosis, the gamete type is capital S, small s, gamete, then small c, small s, then small c, small s, then possible random fertilization. You got this capital C, capital S, small s, small s. So the F1 progeny phenotype is colored full as well as the colorless shrunken. 1:1 ratio. Then the parental phenotype, colored full and colored full. The parent genotype is capital C, capital S, small s, capital C, capital S, small s. So after meiosis, you got the gamete genotype, capital C, small s, and small c, small s. Then this capital C, capital S, small c, small s. So possible random fertilization, you got capital C, capital C, capital S, small s, then capital C, capital S, small s, then small s. So F1 progeny phenotype is colored full, then colorless shrunken. So you got 3:1 ratio. So the ratio of spring between non-linked and linked gene. So the ratio of genotype of gametes from a non-linked cross are different from those produced by linked cross. In the case of non-linked cross, the parental capital R, capital T, small t, crossed with capital R, capital T, small t. The gamete formation is capital T, capital R, small r, capital T versus small r, small t crosses with capital T, capital R, small r, capital T, small t. So F1 generation, you got 9:3:3:1 ratio. So cross link is got this capital R, capital T, small t crossed with capital R, capital T, small t. You got this 3:1 ratio. Okay. 3:1 ratio. So the ratio of genotype of gametes from a non-linked cross are different from those produced by a linked cross. So parentals capital R, capital T, small t crossed with small t. Gametes are capital T, capital R, small r, capital T, small t crossed with small t. The F1 offspring is 1:1:1:1 ratio. If there is a linked cross, means capital R, capital T, small t crossed with small t, he got 1:1 ratio. Next, you see about the sex linkage. Sex linkage is where a characteristic is controlled by a gene on an X chromosome. So sex-linked genes can also be said to be X-linked. The X chromosome carries many more genes, more than 800 genes than Y chromosome. The recessive genotypes for a particular traits that are linked therefore occur more frequently in males than in females. So females have a pair of genes governing each trait. If one gene is faulty, then she has a second one to cover for it. However, if a gene is faulty on the X chromosome of a male, then he may not have a second one to cover and is more likely to suffer an X-linked genetic defect. So the common sex-linked triads is first one is color vision. So gene controlling color visions have two alleles, capital N and it's a normal one and small n is a color blind. Then blood clotting. So gene controlling blood clotting has two alleles. Capital N is a normal and small is a hemophilia. So hemophilia is the inability to clot blood and the hemophilia therefore suffer from persistent bleeding if the deficient protein factor needed is not taken. This is a hemophilia, the X-linked recessive carrier mother, so F unaffected father, then the carrier mother, cross mean you got the unaffected son or the unaffected daughter, carrier doctor, or unaffected son. So hemophilia, there are three possible female genotype of a sex-linked triad. Example, hemophilia, and only two for males. The females capital N, capital N, capital N, small n, small n, small n. In male, capital N and small n. So heterozygous female for this triad is called a carrier. She will pass on the defective allele to 50% of her gamete itself and thus 50% of her children. So there are 6 possible crosses for this trait. The parental phenotype is a normal carrier female crossed with the normal male. The parental genotype is X capital N X N and the X capital N Y. So gamete so produced like this. So random fertilization. So F1 progeny, you got this F1 progeny genotype. The F1 progeny phenotypes are normal female, normal male, normal carrier female, and hemophilic male. Thus, possibilities are present in this hemophilia. Then is a non-nuclear inheritance. The DNA is also found in the mitochondrion. It is also found in the chloroplast. The DNA found in these organelles is described as non-nuclear. The mitochondria and chloroplast replicate themselves in a process similar to binary fission. So note that male gamete only pass on a haploid nucleus at fertilization, whereas the female gamete itself passes on a haploid nucleus. So non-nuclear inheritance, therefore non-nuclear inheritance, that is the mitochondrial DNA is by a way of female gamete only. So non-nuclear genes show a non-Mendelian pattern of inheritance.